Joanna is a postdoc with Sue Clark in the Epigenetics Research Laboratory at the Garvan Institute in Sydney, Australia. Her current focus is on the role of 3D genome organization in cancer and identification of mechanisms by which chromatin architecture arises and is maintained. And I want to say a personal thank you, Joanna, for joining us in the middle of the Australian night. I think it's 2-something in the morning, so we really appreciate that. Thank you for being here. We're all very excited to hear from you, and the floor is yours.
Thanks, Joe. So I'd like to first thank Arima for giving me an opportunity to present my work on the 3D epigenome remodeling in endocrine-resistant breast cancer. So breast cancer is a leading cause of cancer-related deaths worldwide, but around 70% of all breast cancer patients are estrogen receptor positive. And these patients will receive some kind of endocrine treatment. And as you can see on the Kaplan-Meier plots, this treatment significantly reduces the risk of relapse. However, there's still around 30% of patients, I'm sorry, close the chat. Yeah. Around 30% of these patients treated with endocrine resistance will relapse within the first 15 years of receiving treatment. And that is due to development of endocrine resistance. And our group has a long-standing interest in understanding the mechanism behind endocrine resistance with a special focus on the epigenome. So in order to look at the epigenetic changes associated with endocrine resistance, we use a cell line model, which consists of ER-positive breast cancer cells, so MCF7s, which are then treated for long-term with either tamoxifen until the development of a tamoxifen-resistant cell population, which we call TAM-R, or with another endocrine treatment called fulvestrant until development of fulvestrant-resistant cell populations, which we call FAS-R. And using this experimental model, our group has previously shown that when we look at the DNA methylation patterns in endocrine resistant, shown here in red, and endocrine-sensitive breast cancer cells, we can see that there's huge hypermethylation in the resistant, in the two resistant cell lines, and that this hypermethylation is associated with loss of ER binding, and that frequently occurs at enhancer regions. So considering this hypermethylation happens at enhancers, my hypothesis when I joined the lab was that loss of ER binding at these enhancer regions, as shown here, which is due to hypermethylation, results in a disruption of local enhancer promoter interactions and then aberrant gene expression in the resistant cell populations. So in order to study this, in order to validate this hypothesis, we'll use in situ Hi-C in the endocrine-sensitive MCF7 cells and two endocrine-resistant cell lines, TAM-R and FAS-R. And we looked at changes in 3D chromatin organization and then compared that to different histone modifications and transcription factor binding sites using ChIP-seq. We also looked at DNA methylation using whole genome bisulfite sequencing, and we looked at changes in gene regulation using RNA-seq. So as Anthony already mentioned, the 3D genome is organized inside the cell nuclei in a very tightly organized manner. And the first level of organization that we looked at were the chromatin interactions. So these are interactions frequently between enhancers and promoters. They're maintained by CTCF and cohesin, and we know that chromatin interactions connect regulatory elements like enhancers to the target gene promoters, and that facilitates correct gene expression patterns. So in order to identify differential enhancer promoter interactions between the sensitive and resistant cell lines, we used a diff Hi-C package. And using this, we identified around 2,000 differential interactions between the MCF7 and TAMAR cells, and around 5,000 differential interactions between MCF7 and FASR cells. Interestingly, we saw not only big differences between the parental MCF7 cells and the resistant cell lines, but also there was high dissimilarity between the TAMAR and FASR differential interactions, with actually just a few common differential interactions between them. We observed both loss of existing interactions, shown in green, as well as gain of novel interactions. Yeah, and as I mentioned, there's only a few common differential interactions between the TAMAR and FASR. So we obviously next wanted to see where these interactions are happening in relation to the epigenome. And we saw that gained interactions were highly enriched for active enhancers and promoters. Shown here, when you can see that lost interactions have lower enrichments compared to the gained interactions in TAMAR and FASR. Considering these are active enhancers and promoters, we looked at the expression of genes located at anchors of these interactions, and we observed that at anchors of gained interactions, majority of the genes were upregulated, while at the anchors of interactions that were lost in resistant cells, we saw downregulation of gene expression. There's still a lot of genes that go in opposite direction, which we haven't really looked at. So here is an example of a GREP1 gene, and you can see in that gene that all those long-range interactions that are present in the endocrine-sensitive cells here are lost in both of the resistant cell lines. And this loss of interactions is associated with complete silencing of gene expression, as shown here on the RNA-seq track. And importantly, GREP1 gene is a very well-known predictor of endocrine resistance, and its loss is associated with worst patient survival in endocrine-treated breast cancer patients. So a key gene that we saw is being downregulated in endocrine-resistant cells is ESR1 gene, which encodes the estrogen receptor itself. So here you can see that its expression is significantly downregulated in TAMAR cells, and it's completely lost in FASR cells. And we know that estrogen receptor binding is significantly remodeled in resistant cells, as well as in clinical samples. When we compared the actual ChIP-seq data between the MCF7 and TAMAR, we saw that there's a huge loss of ER binding and some, as well as some gain of ER binding. So next we looked at the association between the differential interactions we identified and altered ER binding sites. And we saw that loss of ER binding was associated with loss of interactions in tamoxifen-resistant cells, while gain of ER binding was associated with new gained interactions in tamoxifen-resistant cells. Actually, when we looked at the total overlap, we saw that around 30% of both lost and gained interactions were actually ER-bound differential interactions. And when we looked at the gene ontology terms enriched at this key ER-bound differential interactions, we saw lots of breast cancer-related pathways that have been previously associated with endocrine resistance. So our previous work has shown that DNA methylation at enhancers that are bound by ER is associated with endocrine resistance. And so to confirm this result, we now looked at the genome-wide DNA methylation patterns, and we again observed that hypermethylated, differentially methylated regions are associated with loss of ER binding in tamoxifen resistance, while hypomethylation was associated with new ectopic ER binding. When we plotted the DNA methylation data genome-wide at the ER binding sites, we saw a big shift in methylation at these sites. So less methylation at new gained ER binding sites in tamoxifen resistant cells. And importantly, when we looked at ER binding sites that were gained in endocrine resistant tumors from the public data, we saw less methylation in those metastatic tumors in whole genome bisulfite sequencing from metastatic tumors as compared to primary tumors. However, we only have a couple of tumor samples to look at. So this association is not significant, but we're looking at getting more samples soon. So this is an example of a region where you can see loss of ER-bound interactions in tamoxifen resistant cells, which is associated with DNA hypermethylation. So again, in MCF7 cells, you can see those big long loops at the NCOR2 gene locus. And these loops are completely lost in the tamoxifen resistance. And if you look at the location of anchors of this long lost loops, you can see that this is associated with loss of ER binding in tamoxifen resistant cells at all of these loops. And if you zoom in into those methylation at these locuses, you can see that in each of those differentially methylated, you can see differentially methylated regions at these anchors. So here on those zoom in pictures, you can see DNA methylation track for the MCF7 and TAMAR cells, as well as the whole genome bisulfite sequencing from primary and metastatic tumors, when you can also see this hypermethylation occurring, at least in some of the samples. We also looked at another layer of 3D genome organization, which is the A and B compartments. So these are chromosomal compartments that are either active or which we call compartments A, or silenced. And this, yeah, so compartments A are associated with high levels of transcription and open chromatin, while compartments B are usually located on a periphery and are associated with gene silencing and closed chromatin. So we looked at the association between compartment changes and differentially expressed genes. So we found that around 2% of compartments switch between either from A to B or B to A between the sensitive and resistant TAMAR and FASAR cells. And we actually saw similar number of A to B and B to A changes in the resistant genomes. We observed that these changes in compartments was significantly associated with a change in gene expression, where we saw that if a compartment changed from active A to inactive B, we saw a general downregulation of gene expression. And if a compartment changed from B to A, we saw upregulation of gene expression. So here is a visual example of such a region that changes from active in MCF7 cells, shown here, to inactive B compartment in TAMAR and FASAR cells. And this change is associated with loss of expression of multiple genes within that region, as shown on the RNA-seq track. So we also wanted to see what's the association between the compartment structure and the ER binding profiles. And we saw that if a compartment changed from A type to B type, we saw general enrichment for lost ER binding sites in resistant cells. And when the compartment changed from B type to active A type, we saw enrichment for gain of ER binding sites. And again, just to show you a representative example of that, here is a region in MCF7 cells that's inactive and compartment B. That region becomes active A compartment in Tamar cells. And on the ER ChIP-seq track, you can see a huge increase in ER binding at that gene locus. So in conclusion, we have shown that 3D epigenome remodeling is a key mechanism underlying endocrine resistance in ER-positive breast cancer. And that includes differential ER-bound enhancer promoter interactions, which are associated with aberrant expression of key breast cancer genes. They're located at altered ER binding sites in resistant cells, and they are associated with DNA hypermethylation at these sites. Additionally, we saw that a change in A and B compartment structure, which occurs at differentially expressed genes and at altered ER binding sites. And with this, I'd like to thank my lab with PI Susan Clark at the Garvan Institute and all the people who contributed to this work. Thank you.
Thanks, Jo. We have several questions here. Let me start with Shara Lampos asks, what causes the changes in DNA methylation at the sites that you presented?
I would think that these sites are not being, so we know that ER binding changes in that the ER can be kind of shuffled or pushed around on the genome to different sites to activate different genes. And we think that DNA methylation is kind of the final mark that closes the chromatin at these sites and makes it inaccessible, and that's why we lose the looping. So it's kind of a closing mechanism for these sites.
Okay, yeah, I think that there was a related question to that that maybe you touched on a little bit is, Papudu asks, can you say whether these changes and interactions that you observe are causal, or are they results of other dynamics like DNA methylation or histone modifications?
Yes, I think that's a very good question. So we would like to think that it's possibly that the epigenome has to change first. So either the DNA methylation has to be gained or lost, or the chromatin, like the accessibility has to be changed for the loops. And we do actually have a temporal model of endocrine resistance in which we plan to check what actually happens first. But I think everything happens very, very fast, which means that we would need very fine temporal data to be able to tell whether it's gene expression that happens first, accessibility, DNA methylation, or looping. But yeah, that's definitely something I'd like to check.
Okay, thank you. Sabria asks, do you have evidence of estrogen receptor associating with mediators of promoter enhancer looping, like mediator, complex, cohesin, or at least sharing common binding sites?
So I haven't looked at it myself because I don't have the cohesin. I have cohesin data for the cell lines, but it's not as, so we don't get many peaks, and it's not very good quality, and cohesin is quite difficult to chip. I know that ER and promoter enhancer looping is associated with different chromatin remodelers, which has been shown previously by Jason Carroll's lab. But I haven't looked exactly at cohesin and the association. I think that's mostly because it's not such a good chip as CTCF, for example.
Okay, there's another question here that I guess I was thinking of as well. Jihad asks, have you validated these types of findings in primary tumor tissues?
Yes, so this is where our collaboration with ARIMA comes in. So we do have endocrine-resistant PDX tumors, which were implanted in mice. We have a couple of different PDXs, and we are currently working with ARIMA to use the low input method on fresh frozen tumor tissues to see if we can see the same kind of changes, at least in PDXs for now, because that's where we have most of the tumor tissue available. But if we get the method working, we would like to move to actual patient tumors, which we do have for our collaborators at the King Kong Cancer Center in Sydney.
All right, maybe just one or two more technical questions about the analysis itself. Subhashri asks, how do you identify different compartments A and B from the Hi-C data?
So I know there's a couple of different methods to identify compartments. For this work, we used Homer, which was the easiest one because it just works on mapped reads files, so BAM files. So these are just PCA kind of analysis, but I know that Juicer as well has now eigenvalue compartment calling. So there's a few different methods. Yes, but in this work we used a software called Homer